comes to your mind when you hear about temperature and enzyme? Very fantastic question, right? What comes to your mind? Welcome to another lesson on BET. Biologists explain this. We'll be explaining today or discussing or shedding more light on factors that affect enzyme action. Okay. Remember, so far in our lessons previously, we've discussed issues regarding the shape or structure of enzymes. We've also discussed what happens in the active site, the concept of catalysis, which we said has to do with stressing or weakening bonds that hold the substrate molecules together so that they are freed now to recombine and form products. Okay, so today we are discussing, because before we rounded off last session, we listed, or I told you or prepared your mind about factors. So let's do a quick recall. One, we talked about temperature. Two, we talked about pH. Three, we talked about the substrate concentration. And four, we talked about, or we mentioned, enzyme concentration. And five, now we are discussing, or we mentioned, inhibitors. So today we will be discussing temperature briefly. Now, the misconception in the heart of many students is the fact that every time the issue of temperature is mentioned, you just quickly jump at it. Wow, the enzyme is destroyed. Now, let's first of all understand this. Another problem is the idea that an enzyme is killed. Come on, it can happen. An enzyme is not a living thing, like we've said previously, it's just a protein structure. So you can't kill an enzyme. Please understand that, dear listener, it's you can't kill an enzyme. What you do is to distort or to damage or to destroy what we said previously was the three-dimensional shape of the enzyme that makes the active site to have a unique shape that complements the shape of its substrate molecules. So what you do is that rather than jump at the conclusion that any temperature that you are given is damaging, you have to be very careful. Let me say that again. The habitat an organism lives determines its temperature range. What if I told you, for example, that there are some organisms called archaebacteria? One of them is Thermus aquaticus. Now, these bacteria, they live in what the places you call the hot springs of the world, and they thrive at 110 degrees Celsius. Read my lips again. Thermus aquaticus and several other bacteria, you call them extremophiles because they live in very extreme conditions, very low pH, very acidic, extreme temperature of 110 degrees Celsius, even above boiling temperature, and they are not killed, meaning they are adapted. And there are animals in deserts, for example, very hot in the daytime, very cold at night, and they are thriving. So you have to understand that temperature doesn't just necessarily have to be damaging. So let's look at the temperature in context. Now, if you look at temperature, what you should have in your mind is this. Discuss temperature from three perspectives. Okay, let me say it again. Three perspectives. One, low temperature. Okay, so you go from lower temperature and then you go gradually to what you refer to as optimum temperature, and then you exceeded the temperature and then you have beyond the optimum. So let's look at again: lower temperature, optimum temperature, and beyond the optimum. Now quickly, let's look at each of these three. At lower temperature, which is what is done when you refrigerate anything. Now low temperature doesn't kill, but it inactivates enzymes. Now if you have let's say you have a contaminated beef or pork or chicken meat, for example, and you manage to refrigerate it, what you have done is that even though the meat or the pork is contaminated, the bacteria that you have there, they will not be able to thrive because the temperature makes them damn uncomfortable, very uncomfortable. So they will be incapacitated. But the moment you bring out this meat or pork at temperature and the meat begins to thaw, suddenly the temperature begins to get warmer and warmer, and unknowingly to you, these bacteria are becoming very comfortable. So if you don't cook your meat or your pork properly, you can get infected with the bacteria, leading to disease conditions. So low temperature incapacitates, makes these microorganisms very uncomfortable. Their enzyme will not work at lower temperature. Okay, now as you begin to increase the temperature, the enzymes, like we said, enzymes are made active by increasing temperature. So in your mind's eye, begin to see this, that lower temperature inactivates, making enzyme molecules to be passive, as it were. But as you increase the temperature, remember, increased temperature increases the kinetic energy of any molecule. And so proteins are molecules, enzyme proteins are molecules, and then if you look at the substrate themselves, they're also molecules. So both the substrate molecules and the enzyme molecules, they become more active. So there'll be more collisions leading to more enzyme substrate complexes being formed, and so products can be formed. So what you should remember from this discussion is that increasing temperature increases the kinetic energy of substrate and enzyme molecules, making them more active and hence leading to product formation, because there's an increase in the rate of activity and reaction between the substrate and the enzyme molecules at the active site. So the question is, when you begin to approach the optimum, meaning you have activated these molecules to the highest they can tolerate. Okay, once you get to the optimum, that is the best. Okay, meaning the reaction is working at the best and the most favorable condition as far as temperature is concerned. And so you have enough ES complexes, enough products being formed, and the reaction is just going on enjoying the temperature. The moment you begin to exceed this optimum temperature, and let me say this, the optimum varies. Okay, it's different with different kinds of enzyme molecules. I just talked about Thermus aquaticus, an antibacterium that lives in the hot springs part of the world that can endure above boiling temperature. There are some that even 60 degrees Celsius can damage. So the idea is, for the kind of organism you are discussing, or the kind of enzyme protein you are discussing, it doesn't matter the optimum. Once it's exceeded, okay, bonds are broken. Remember, there are four kinds of bonds that maintain the tertiary structure of a protein molecule. Four. You've got the weakest of them, hydrogen. Then you've got ionic bonds. Then you've got van der Waals forces, what you call hydrophobic interactions. And the strongest of the four bonds, disulfide bonds. Okay, so let me take that again. You've got hydrogen, you've got ionic, and then you've got van der Waals forces or hydrophobic interactions, and fourthly, you've got disulfide bonds or interactions. So that's why bonds are the strongest. That's the extreme end, and then you've got the hydrogen as the weakest. So any bonds that you are able to break by exceeding the optimum temperature, you have distorted the shape of the active site. Okay, that is what we refer to as denaturation. De means as a prefix to remove. Remember, you can detoxify, remove a toxin. You can decarboxylate, remove carbon in the form of carbon dioxide, CO2. You can dehydrogenate, remove hydrogen. So denaturation is to remove the actual nature or the form of that protein molecule of the enzyme. So when you're frying egg, for example, it's a kind of denaturation. The albumin and the yolk were liquid, and then as you are frying, they became solidified. That is denaturation. So picture that in your mind's eye, that when you the issue of denaturing a protein molecule occurs when the optimum temperature is exceeded. Like I said, don't conclude, don't rush into that idea that every time temperature is mentioned, it automatically means you have denatured the enzyme protein molecule. And remember, you can't kill enzyme because they aren't living things. And so the habitat of that organism determines its optimum temperature. Some 60 will have a problem. As human beings, as we are, for example, we have what we call 37. One is 37 degrees temperature. That is what we call room temperature, what is favorable. In fact, in plants, majority of plants thrive at 25 degrees Celsius. Okay, apart from the ones that you call the C4 plants, okay, that live in the tropics, where temperature is higher in the environment. But remember, we have been discussing enzymes as biological catalysts that function within cells, not in the environment. And so the temperature of the forest or the desert will not work within the body. There are systems, when we get to the topic of homeostasis, we'll be discussing the issue of the fact that homeostatically you have to moderate what is going on in the body. And so there are processes, there are mechanisms for ensuring that for any factor or condition the cell finds itself, there is a moderation. And so remember, low temperature, optimum temperature, and beyond the optimum. And remember the idea of denaturation. That rounds off our lesson for today. I hope you enjoyed it. Now, I'll be sending you questions to try to apply the concepts that we just discussed today. See you next time. Bye.